professor, physician, epidemiologist. I am Dr. Sri Banerjee. Hello, I'm Dr. Sri Banerjee, epidemiologist, biostatistician, faculty member, and in this video I'm going to be going over how to use SAS to calculate relative risk. So let's get started here. So I'm going to start with the code prototype. This is actually a specific segment of code that you can use to upload onto the on-demand academics website. And that's the cloud-based, server-based place where you can compile your code and see the output. So this is a sample code. The nice thing about this piece of code is that it doesn't have to be attached to any sort of data set. So it actually runs more like a calculator than something that actually is doing statistical analysis. So let's go through the steps of how this all occurs. So in this segment of the video, I'm going to be going over an article that was written, a very important article in regards to the efficacy of the COVID-19 vaccine. So this was looking at Moderna and Pfizer and just how effective the vaccines were. So here, my objective here is to show you how to calculate a relative risk of getting the COVID-19 infection for if you had the vaccine versus if you did not have the vaccine. So and this was in the assignment. There is important information about how to get subtype analysis through females, but in this example, what I'll go through is look at subgroup analysis for males, and give you kind of some pointers on how to determine this. So what you can do is when you're trying to look at the number, you take this number and then you take a look at how many people develop the infection. And so this is how you calculate it. So the first thing we want to look at is exposed disease. So exposed disease is for males, and remember at the beginning I said that I'm looking at males only here. So exposed disease would then be 3. So that's the first number you need. And then let's take a look at the second number. So the second number, not exposed, diseased. So that number is, so this is exposed and not diseased. So this is exposed and disease, and then I'm not going to write the second line, but it's exposed, not diseased. So that is 8, 8, 7, 2, because we subtract the 3. So that should be your second line. And then your third line is not exposed, disease. And then you have not exposed, not diseased. So that actually would be. So these are the four main numbers that you need. And now that you have these numbers, now you're ready to place them into the prototype code, and then put it into SAS. So I'll show you how to do that. So in this part of the video, I'm going to show you a systematic way to not only do some coding, but show you how you can apply the data mining principles to use the numbers that you have derived from the clinical trial for COVID-19 vaccines, and then calculate some interesting relative risks of developing COVID-19 in males if you did receive the vaccine versus in the placebo group. So before I look at the calculations and the numbers that we mined from the last section, what I wanted to do is revisit the prototype code that we first started off with. So the prototype code has been put in place, and you can run the numbers and see if it actually gives you some relative risk and odds ratio. So definitely does give you some measures of association. And these are the values that you are most interested in terms of understanding the measure of association, and the calculation. Um, so here is some output data. And then now what we're going to do is we're just going to highlight, and this is a good way to practice for yourself as well, is we're just going to highlight the segment that we want to compile. And so when you want to do that, all you got to do is highlight and then select run. And this is run for SAS. And so just a step back from this, in order to get into the system, the online cloud system, you have to, of course, create an account, which you would have done in a previous week, and then just copy and paste the code onto the layout. And this is the command for the new program. So if I select this new, then SAS program will open up. So that's how you do it, and then you just copy and paste. But going back to compiling just this segment of the code, let's go ahead and do so. And if we do so, we'll see that the new numbers, and this is really neat and interesting, because now you've calculated, this is what you've done. You've actually gone and calculated the relative risk and odds ratio from an actual clinical trial, published clinical trial. And here is your output data. And so always get into the habit of looking in the log and making sure nothing went wrong. There's no red in here, so that's always good. So there you have it. That's all the different facets of running your own code and providing information from clinical trials. Now you can go into any clinical trial and get these numbers from that using SAS. Thank you for listening.